Good morning, good afternoon, or good evening, everyone, from wherever you're joining us, and welcome to today's cutting edge conversation titled Addressing Orphan and Rare Diseases. I'm Sarah McFarland from the events team here at scientist.com, and I'm really happy to be your host for today. And now I'm going to just give a really brief overview of our topic today. An orphan or a rare disease is a condition affecting a really small percentage of the population. In the United States, a rare disease impacts fewer than 200,000 individuals, while in the EU, it is defined as affecting fewer than 1 in 2,000 people in the general population. To illustrate this, imagine this grid with 2,000 squares is actually 2,000 people, and only one square there in orange would represent those affected by a specific rare disease. Significant strides have been made both in the US and internationally concerning rare and orphan diseases. The Orphan Drug Act, established by Congress in 1983, coined the term orphan diseases due to the pharmaceutical industry's lack of interest in developing treatments for such small patient populations. The Act introduced financial incentives to encourage the development of new drugs for these conditions. Similarly, the EU implemented Regulation 141 in 2000, known as the Orphan Regulation, which parallels the US initiative. There are an estimated 6,000 to 8,000 rare diseases, collectively affecting 25 to 30 million Americans. This highlights the paradox that while individual rare diseases are uncommon, the likelihood of knowing someone with a rare condition is actually relatively high. Examples of rare diseases can be seen here and can result from infections, birth defects, cancers, or genetic conditions. But many remain untracked, which complicates prevalence estimates. So what's being done? Efforts to address these challenges are ongoing. In the US, the NIH operates the Genetic and Rare Diseases Information Center, which focuses on research and awareness, and Rare Disease Day is observed on February 28th each year to raise awareness. Internationally, the EMA has its own framework, while the WHO prioritizes research needs for rare diseases. The United Nations has also adopted a resolution advocating for people living with rare diseases, all of which signals a commitment to advancing research and support over the next 2 decades. Today, we have 2 experts here to discuss how their companies are working in this space. I'd like to thank both of these sponsors, Health Lumen and ICON, for their contributions today and for making this event possible. And to kick things off, I'm very pleased to welcome Dr. James Cook to the floor. James, please take it away whenever you're ready.
Great. Thank you so much, Sarah. So yeah, I'm James, and I'm going to be talking today about how we quantify rare genetic disease patient populations using genetic databases. So I want to start the talk today just with a question, and that is, what is the true scale of rare diseases? As Sarah's already mentioned, her numbers were 6,000 to 8,000. Ours are a little higher, 7,000 to 9,000 rare diseases that have been described to date. And these have major physical and emotional burdens on the individuals and the families who suffer from them, as well as emotional and societal burdens more broadly. So we often use the term individually rare, collectively common. It is not uncommon to know people who've got these diseases. It's estimated that 95% of these diseases currently have no approved therapeutic, although with things like the Orphan Drug Act, that number is coming down all the time as improvements are made to the care of rare disease. And one thing that does make it particularly difficult to study and to develop treatment for rare disease is just getting a handle on the prevalence. There are wide ranges of prevalence estimates in the literature. These typically have been based on reported outcome data. So this really only gives a partial picture of those people who've been diagnosed and known to the healthcare systems. So as such, these numbers are often incomplete and can underestimate the true scale of rare disease. And the advent and the availability of large genetic databases offers new possibilities and new methodologies for closing the data gaps and generating more robust prevalence estimates using what we call a bottom-up approach. So I just want to kind of come on to why this matters. And really, it's because these numbers inform every stage of the molecule to market life cycle. right from kind of the early stage R&D, so understanding disease etiology, identifying disease targets, specific mutations which may be amenable to therapeutic intervention, all the way through clinical trials and identifying and stratifying patient groups for clinical trial recruitment, HEOR, market access and launch, so generating budget impact forecasts and HTA dossiers, after launch, post-market surveillance, market expansion, the identification of additional populations, additional genetic variants potentially that could benefit from the same therapeutic, and finally policy impact and patient advocacy, so helping with the case to raise awareness, which helps to drive change and improve the knowledge and understanding of these conditions. So what we have here is our iceberg diagram. So we have the proportion of the iceberg above the water, which is our known population ascertained by metrics such as hospital case diagnoses, physician or clinician diagnoses, or people who have a known genetic risk for a condition. But there can often be this larger unknown population, which makes it very difficult to quantify the patient population. So these can be people who've been misdiagnosed, undiagnosed, people with limited access to the healthcare system, those who have an unknown risk, say if they carry a genetic risk factor unknowingly, and poor health system infrastructure as well. And this sometimes can be a little bit more complex than just a poor health system. If you take something like the ICD-10 coding system, which typically is used to record diagnoses within medical records, many rare diseases don't actually have their own distinct code. So it can make it quite challenging, even if you have a correct code, to work out exactly what the condition is that's being diagnosed. And the idea of the work that we and others are doing is to try and raise the iceberg so that there's a greater proportion of the population of a rare disease which are known to the medical system. And I just want to present an example here. So this is just looking at 10 lysosomal storage disorders. And these can be broadly grouped into two sets of 5. So for each of these diseases, we're presenting a range of prevalence estimates. The top 5, it's prevalence estimates within the same or within very similar populations. And the bottom 5 is prevalence estimates between different populations. And I think it helps to really illustrate the issue. So if we take something like in the top half, Krabbe disease. So within the US, this has an estimated prevalence of around 1 in 310,000 to 1 in 20,000. So that's a 15-fold difference in estimated prevalence. Whereas looking towards the bottom, that Fabry disease, for example, around a 2 order of magnitude difference in prevalence between Australia and Taiwan. And getting a handle on the prevalence is really important when it comes to planning how to develop and how to release therapeutics. So our work to quantify the symptomatic population starts with the identification of causal genetic variants. So we analyze data from genetic databases to identify variants which are associated with disease, or if they haven't been formally associated with the disease, with consequences within a gene which would be expected to cause the disease. We assess the literature to review the existing knowledge, in particular looking into populations which have high disease rates. Rare diseases can typically vary widely, as we've already seen, between populations. We then use this information to quantify carrier rates. So we extract our allele frequencies, establish a baseline of expected carrier counts in diverse global ancestry groups. Where data is available, we look to extract allele frequencies for specific populations where the disease is endemic, although this data is typically quite hard to get hold of. We then apply penetrance data. So penetrance is a measure of the proportion of the genetic carrier population who would be expected to become symptomatic. So if you apply the penetrance rate to your overall carrier population, you can calculate from that how many actual patients you may end up with. We use a wide variety of databases to do this research, but I do just want to highlight a couple of examples. So we use data from the Genome Aggregation Database, or GNOMAD. So this contains allele frequency data and functional annotation for tens of millions of genetic variants. And this uses data that's contributed from around the world. So this is used for calculating carrier counts of specific genotypes in diverse genetic ancestry groups. And this can be combined with demographics data to project allele frequencies into population data and to calculate carrier counts in countries of interest. We also use specific biobank level resources. The example I'm using here is All of Us in the US. which is a research program run by the National Institutes of Health. And this contains linked genotypic and phenotypic information for a large sample of citizens within the US, with a specific focus on trying to reach populations which haven't previously been studied as rigorously as others. And we use this kind of database for examining the relationship between genotype and phenotype. So, for example, we can identify a cohort of people carrying causal genotypes for a disease and compare their medical records with non-carriers to assess a range of things such as penetrance, symptom severity, age of onset of disease, etc. And what we're looking to do here, we can kind of describe by us peeling back the layers of the onion. So starting with the outer layer of the onion here, we have a population carrying a causal genotype for disease X. The layer within that is the layer that the people who are affected by symptoms. A smaller layer, again, will be formally diagnosed with the disease. Within that group, a subgroup will be amenable to treatment and therefore fundable by the healthcare system. And some of those will be eligible for clinical trial, depending on what is the aim of the company that wants to perform the clinical trial. And it's important here to really state the role of screening here, which aims to increase the proportion of those affected by symptoms who are formally diagnosed with a disease. And that will then drive the size of the population group amenable to therapy. And I've already mentioned that these diseases vary kind of wildly between different populations. I want to point out a couple of examples here of diseases which can be really concentrated in specific population subgroups. So there are 5 listed on the slide, just to mention a couple: Tay-Sachs disease within the Ashkenazi Jewish population, and Norbottnian and Gaucher disease, which is a subform of Gaucher disease, I think pretty much exclusively found in this region of northern Sweden. And to help with this kind of work, we have developed and released a genetic variant prevalence projection tool, which we call Allele Atlas. And this enables population level estimates of the number of individuals carrying causal alleles for rare genetic diseases. So what this does is it extracts data from Nomad, determines the frequency of rare genetic variants in global ancestry groups individually or summed, and then applies national population data to estimate the allele frequency and the carrier counts within the current and future populations where data is available to support that. And this was developed as a free resource for the rare disease community. Anybody can try this out. The URL is at the top of the slide. So yeah, we think this is a really useful tool to be able to assess the genetic carrier population for specific genes which are known to cause certain diseases. So I just now want to touch on a couple of examples of where we've applied this kind of research. And the first is to determine the prevalence of late onset Fabry disease in the US. So Fabry disease is a lysosomal storage disorder. This is an X-linked condition. It's caused by pathogenic alleles in the GLA gene on the X chromosome. So you would expect the carrier count to be roughly double in females what it is in males, because females have 2 copies of the X chromosome. The disease manifests in 2 forms. There is a classic form which presents in childhood and is typically very severe, or there is a milder adult onset form which is less well understood but is becoming more and more studied. And it traditionally has been very challenging to accurately determine disease prevalence. So there were a wide range of estimates available, ranging from around 1 in 40,000 to 1 in 170,000. So we applied our methodology to generate more accurate prevalence estimates for Fabry disease in the US in 2024. And we only looked at 8 selected variants of potentially hundreds across the gene that could cause the disease. But even so, when using reported penetrance figures of 100% for males, so all male carriers will be expected to develop symptoms, and 70% for females, so 70% of carriers will develop symptoms. We actually calculated that there were around 12,000 male carriers, or around 1 in 14,000 in the US, and just under 25,000 female carriers, or around 1 in 7,000. So the genetic carrier population there, compared to the 1 in 40,000 to 1 in 170,000 estimated by the literature previously, we're calculating at about 1 in 10,000. And in fact, there are other sources such as newborn screening studies, which estimate potentially even higher rates than that. So a second example here, we worked with Rally Bio, which is a clinical stage biotechnology company, and they were interested in understanding the size of the population affected by fetal and neonatal alloimmune thrombocytopenia, or FNAIT for short. And FNAIT is a really nasty immune disorder, actually, that occurs when a pregnant woman develops antibodies against platelets from her own fetus. And this can lead to miscarriage, brain damage, or death of the child. This is a really nasty condition, which had previously only been looked at in white Caucasian populations and wasn't believed to be an issue in other populations. So the risk factors genetically are two genetic variants. So there is a form of the HPA locus alone, or in conjunction with a form of the HLA locus. And if a woman carries the HPA-1b variant alone, that is at risk of having a pregnancy affected by FNAIT, whereas if it's in conjunction with the HLA variant, that is a high risk. And I think that's something like a 40 times higher risk than just having the HPA-1b variant alone. So we use data from NOMAD and the US National Marrow Donor Program to determine allele frequencies for the two variants in diverse ancestry groups in the US. And the result was that we demonstrated for the first time that women not of Caucasian descent also carry these variants and are at risk and high risk of developing FNAIT. So we were able to estimate that over 30,000 pregnancies in North America and major European countries were at higher risk of FNAIT annually. Excuse me. And that represented a 40% increase compared to previous estimates. So just want to finish here by touching on some key takeaways. So firstly, obtaining reliable, robust prevalence figures for rare diseases can be very challenging. Published estimates vary substantially between populations, and diseases may have subtypes with limited epidemiology available. And really, the more subtypes a disease has, the more you can break it down, the more likely it is that you're going to run into a subtype with very limited or no epidemiological evidence published. The calculation of reliable prevalence figures is incredibly important for informing pharmaceutical development. It is essential to accurately estimate the market size. And this is necessary both for companies developing products to know what the market size is, and for healthcare payers looking to understand what the burden of disease is within the population. So we've been using genetic databases, which can be used to generate more robust estimates of disease prevalence. So information from global sources can allow the study of rare disease genetics, even in populations which haven't been previously studied and haven't been specifically recruited for that kind of study. And using different types and kinds of genetic databases, we can perform different kinds of complementary research. So I'd just like to finish here by outlining a few of the collaborations and partnerships. So we work with small biotechnology companies, large pharmaceutical companies, as well as in the private, as well as with the public sector, on a wide range of common and rare diseases and risk factors. I just want to say thank you very much, and looking forward to the Q&A later. Thank you.
Thank you so much, James. Fantastic presentation. And now I'm very pleased to welcome Dr. Patricia Kuchaki. Please take it away whenever you're ready.
Hi, everyone. First, I want to thank you for attending this webinar today. I'm very pleased to spend a little time talking about patient experience data. It's really the first step to finding treatment options. And so without further ado, I'll go forward here. One of the important things to think about when you're conducting patient experience data is it's important to consider the context or the lens through which you're collecting the data. In today's talk, I will mainly focus on collecting patient experience data through a measurement lens, collecting patient experience data for the purpose of developing or selecting outcomes measures. But I also think it's important to spend just a minute talking about some of these other lenses because they are also important. The psychosocial lens, with this lens, patient experience data is, excuse me, collected on the social and emotional toll that rare diseases take on families. Because rare diseases are rarely just a disease for that the patient experiences. It's really a family affair. There are difficulties the families face in getting information, getting diagnosed, finding support. there are many other aspects of psychosocial within the psychosocial lens as well. There's also the economic and social determinant of health lens. With this lens focuses on how poverty and lack of access to healthcare resources, educational resources, where rare disease families and patients live, and their social connections and how they impact the health and well-being of the rare disease patient. Then there's the healthcare system lens, which focuses on things like getting a diagnosis for a rare disease or navigating the healthcare system and the financial burden that these issues that they face can cause. Rare disease patients and families tend to require a lot of healthcare. They are involved in a lot of healthcare utilization, which translates into a lot of expenses for these families, and it's really related to difficulties that they have in navigating the healthcare system. Education, this lens focuses on finding information to help families understand and cope with the rare disease. And it focuses on issues related to obtaining sufficient and appropriate educational opportunities for rare disease patients. To educational experiences to meet their unique needs. And then finally, there's the coping mechanisms and AIDS lens, which really focuses on how do rare disease patients and their families cope with rare diseases. What are some of the physical measures that they can take to cope with disease? What are they doing that's helping? How do they help themselves, really? Okay, and these lenses really apply to all diseases, but they're really critical and of major importance, and they're amplified, I would say, in the rare disease space. Okay, well, what is patient experience data through the measurement lens? Well, the FDA's 21st Century Cures Act provides some definition, and basically the focus is on symptoms and impacts of disease, treatment experiences, understanding what's important to patients, and patient preferences. So that gives us a little bit of information on how what the patient experience data is through a measurement lens. The FDA has also issued, and many of you are probably aware of these, but there are 4 patient-focused drug development (PFDD) guidance documents that have been recently published that provide a wealth of information and guidelines on conducting patient experience research. So why do you want to collect this kind of data? Why do you need patient experience data? Where does it fit in? Well, first of all, it fits into the clinical outcomes assessment endpoint selection or development to use in rare disease, and that's the main focus of my talk today. But this information is also important for improving diagnosis, regulatory submissions, reframing patient needs, because what you might assume is a need for patients may not, it may require some reframing to really understand the essence of the problem. Clinical trial design, HTA submissions, and educational material development. And again, these are the patient experience data here is important not only for rare disease, but all diseases, obviously. Okay, now why do we need this patient experience data? I have a very simplistic model here that I'd like to share, and basically, what it is saying is that what we need to understand is what are patients' unmet needs in rare disease. This is at the heart of the matter, really. It basically says that you need to understand unmet needs, what is needed, and identify appropriate technologies that can meet those needs. In other words, what is needed and what is possible. To understand whether the technology can meet those needs safely and effectively, we need measurement tools to understand whether the technologies meet the patient's unmet needs and whether they are safe and efficacious. And I want to point out that lack of patient experience data is really a major hurdle for most rare diseases. This is, they really, there's just a lack of this data to get started. So that, and one reason, and this is obviously true for all diseases, there can be a lack of patient experience data. But many patient advocacy or support groups don't have the funding or the expertise to collect this data. And they aren't really familiar with how to collect the data through the measurement lens that's required to develop and or select clinical outcomes assessments or digital health technologies that can be used to measure safety and efficacy. But at ICON, our patient-centered outcomes team and our Center for Rare Disease are working together to explore ways to help remedy this situation. Some of the information I want to talk about is very pragmatic in a way, but I think it's very important. Collecting patient experience data, where can you do this? Well, you can get it remotely. In other words, this really became very popular in the COVID era when interviews or focus groups were conducted by video conference, and also telephone interviews have been around for quite a long time. And there's also a possibility these days for online panels where patients can meet together as a group, or they can go on and off and add information to the online panel as they would like to do, or when they have time to do it. Then there's also in-person research. This is a very traditional way, obviously, conducting focus groups or interviews at a research facility. But it can go beyond that. It could be conducted at a clinical visit, or many advocacy groups or support groups with rare disease patients attend conferences. And either they have an individual conference for their particular rare disease, or they attend bigger conferences. And that's an excellent opportunity to meet with some of these groups to collect patient experience data. And then finally, just a little bit about in-home visits. This is a particular passion of mine. I can almost guarantee that if you do in-home visits with patients and their families, you will never forget those visits. I know I can remember some that I have done. Obviously, in-home visits require more, you have to overcome the confidentiality issues and so forth. And it can be more expensive and time consuming, but you really get to know more about what the living situation for rare disease patients look like. And if for instance, if they have certain aids or coping mechanisms that involve something that is in their home, they can actually show you that. So I really have a passion for this, although I know that it does have its limitations in terms of time and money. Okay, and so who do you collect patient experience data from? Well, it can be collected from physicians, caregivers, and patients. And all of these sources of data can be used to understand patient experience. And as many of you probably know, when data is collected from physicians, that can help lead to the development or the selection of clinical reported outcomes. If it's a caregiver, if data from a caregiver, that can be used to develop or select an observer-reported outcome. And if the data comes directly from the patient, it can lead to a patient-reported outcome. So all of these data sources are important, particularly caregiver or observer reported outcomes are particularly important because oftentimes patients are children and they may have limited ability to communicate. So all of these sources of information and the outcomes measures that come from those, from the experience data, those are all important. I wanted to mention one particular example that about two novel COAs that contributed to the approval of a new drug for PFIC. The patient experience data showed that pruritus associated with this disease can be very severe and disruptive for patients and families and limits patients' ability to participate in daily activities. One of the most prominent symptoms of PFIC and signs as well relates to unrelenting itching, scratch marks, excoriations, and scarring due to persistent itching. And interestingly, in this situation, an OBSRO and a PRO were developed to assess the severity of pruritus on a 5-point Likert scale using an electronic diary. And the OBSRO actually measured the signs of pruritus, such as scratching. And as you can see in the diagram, there was a 5-point Likert scale that was related to the scratching activity. At the same time, a PRO was developed for patients to complete, and it too had a 5-point Likert scale. And it was focused on the activity of itching, so that interestingly, that the OBSRO was the primary endpoint in this drug approval, and the PRO was the secondary measure. So that sometimes it's important to develop multiple or use multiple clinical outcomes assessments to understand or to measure a particular disease in clinical trials. Okay, I have a few other considerations that I'd like to mention. We typically, and I've already alluded to the fact that the primary focus for COA selection or development is focusing on symptoms and impacts of symptoms on patients' ability to function, perform activities of daily living, and overall health-related quality of life. These are the treatment goals, the unmet needs. However, there are some things that have to be considered beyond just the very, the central theme of symptoms and impacts. For one thing, the severity of disease with disease progression and disability trajectory. Some diseases, and I have an example here, Duchenne muscular dystrophy, as the disease progresses, the situation and the patient experience differs. So basically, the experience has been divided into 3 periods: ambulatory, where patients are still mobile; a transitional period where they're sort of in between and they're having some difficulties with mobility; and then finally progressing to a stage where they are non-mobile. And so what's important, and this study in particular pointed out that what's important can vary based on where you are along the progression scale, along the disease progression. So that you have to consider when you are developing or looking at a technology and you want to measure its effectiveness, you have to consider which part of the progression, disease progression. Where are you along that progression? So that's one thing to consider. Also, you have to think about these adaptations and coping mechanisms, and how they impact how patients are perceiving their unmet needs and treatment goals, because they could have a masking effect of some of the, if they're able to do things because of these adaptations or coping mechanisms, then they may not seem as important to patients as they would be if they were thinking about the symptoms without these mechanisms. Yeah, and the evolution of the burden of disease on patient and caregiver. In other words, this too can change over time. Um, the requirements for support and so forth can vary, and it's something to think about too in terms of measuring what's going on um, with patients. And since most rare disease patients are children, disease progression happens in the context of natural childhood development. In other words, children are getting older and changing naturally, but then the disease progression is overlaid on top of that normal childhood development. So sometimes it's important to think about that. Also, COA endpoints for children versus adults with the same rare disease, for example, osteogenesis imperfecta. What's important to measure for children can be different in adults. In the case of OI, in children, historically, the fracture rate has been an important endpoint for measuring success, measuring efficacy and safety for children with OI. However, adults don't experience fractures nearly as often, so that regulatory agencies have been expecting that clinical trials, the endpoint that would be used in adult trials would be the same as for children. And so clinical trials where the endpoint has been fractures have not been very successful, because that's not really what's important to the adult patient. The adult patients have deal with pain and muscle issues, and so that the endpoints for the adult patients should really be different than for children. And sometimes there is resistance from regulatory agencies to want to change what they've always done. And so patient experience data is collecting it for the adult patients in this case would be very helpful, or should be very helpful. I'm not saying that it has been. It's always a bit of a battle, but at least the patient experience data should point in the right direction as far as regulators go. Okay, and now collecting qualitative patient experience data in rare disease, how do you collect it? Well, you can collect it longitudinally, and this is how patient experience data is often collected in natural history studies. And basically, what you're looking at is what is patient experience over time, and it's very useful for understanding disease progression. And you can follow the same patient population over time. However, it's expensive because it is over a longer period of time. This type of research is subject to patient participant dropout, and it can be very time consuming. The other alternative is cross-sectional research. It's generally less expensive, less subject to dropouts, and it can provide some understanding of disease progression by conducting patient experience research with patients with varying severity levels. So the disadvantage really is that you're going to be looking at different patients, and so at each level of progression, so that it's not quite as clean as following the same patients over time. And I also wanted to say a little bit more about the need to reframe adaptation or coping mechanisms. Um, because, as I mentioned, when patients use aids or coping mechanisms, it can mask the real issue that they have. And so you might want to ask, have you found anything that helps overcome the symptoms of impacts of your disease? And what are these and how do they help? But imagine if you no longer had these things, what would your life be back? What would your life be like? And what things would you not be able to do? And how and why would this be important? So it's worth spending a little time trying to dig beneath the use of these coping mechanisms, because there may be some important insights that you will miss if you don't. Okay, and this is another passion of mine, is having patients or caregivers do some pre-work before patient experience research. And so why pre-work? Well, it really engages the participants in the research before you ever meet with them to actually have an interview or to participate in a focus group. And it also it gets patients thinking about, or caregivers thinking about, the topics that you want to discuss in the research. And it also can be an excellent conversation starter once you have your participants together or once you start your interview. So what are some of these types of pre-works? Well, there are projective techniques where you use visuals, personification, or word mapping. Visuals, what I mean by that is basically having patients or caregivers choose visual representations of what their disease is like and how it impacts them. And it also, or pictures or images that can depict what life would be like or what they would be able to do if they did not have the particular disease that they have. And personification is really, it's just kind of a fun thing, and it really gets patients and our caregivers engaged, asking what type of you can ask anything you would like to personify. It could be what kind of person would you be if you didn't have this disease, and what kind of person are you, or what profession are you in, or even you can ask what kind of animal are you, and what kind of animal would you be if you didn't have this particular disease. Word mapping is really starting with, it's really just a way to develop a map of words that patients or caregivers associate with a particular disease. And so it can be really good in terms of understanding the language that patients or caregivers use. And finally, narratives and storytelling, I think that that's pretty self-evident, but I think that all of these techniques are really useful when conducting patient experience research. And finally, I would just like to present to you uh, some resources that you can look at if you're interested in conducting patient experience research. One particular group I'd like to point out is the Rare Disease Clinical Outcomes Assessments resource, the RDCOAR. This resource is an initiative of the Critical Path Institute's Rare Disease Clinical Outcome Assessment Consortium. And its goal is to really try to help identify clinical outcomes assessments that are appropriate for rare disease clinical trials. And so with that, thank you all for listening, and we'll move on, I guess, to the questions and answers.
Thank you so much, Patricia. Okay, and now I'm going to bring our lovely speakers on. Thank you both again for those fantastic presentations. And if you're ready, we are going to jump right into the Q&A. Okay, so this first question is for you, James. You mentioned that you're able to scale the information from genetic databases to arrive at a population level analysis for specific rare disease. Can you expand on how that works and what sources you use for population data?
Yeah, sure. So we get allele frequencies reported in terms of the number of times a specific allele, so in this case, a pathogenic allele for a rare disease, per 100,000 observed alleles. So that's per, unless it's an X-linked disease, that would be per 50,000 in the population. So we can then take that rate and extrapolate it up to the reported population within a country while mapping genetic ancestry groups to reported demographics groups in a population, whether that's racial data, ethnicity data. And we tend to get that data from national census sources where possible. The US has a particularly good census bureau that reports this data really well with, I think, 50-year projections into the future. In the absence of census level data, we can resort to something like UN data, which has been really useful for us in the past.
Okay, thank you. The next question here, I think, is for you, Patricia. This question is, at the beginning of your talk, you presented a simple model where PED, or patient experience data, is used to understand unmet needs, and technologies used to meet those needs are linked from a measurement perspective. Isn't this model a bit too simplistic?
Yes, I definitely think it is. And I think one of the things that is missing, I think, is understanding the basic, and it sort of relates to what James has been talking about, understanding the underlying mechanism of the disease, because obviously technologies are not, they don't come out of the air. There needs to be a biological or biochemical basis for those. And I think that there's a lot of work going on at the moment to try to understand genotypically what is going on in rare diseases. So I think that that is one thing that's very important. And although I said that there are these individual lenses, I think really some of those lenses do have a direct impact on measurement in some way. So I think that what I would say is, it is more complicated, but for purposes of discussion, I think a simplistic model suffices.
Absolutely, thank you. Okay, the next question here is for you, James. This question is, the recent development of genetic databases has resulted in very powerful sources for epidemiological analysis. Can you comment on future trends? What can we expect in the next phase of development for these databases and biobanks that can be applied to help the rare disease community?
Sure. So there are a few forms of potential expansion for biobanks and for genetic databases. The first always is more sample size. So I mentioned the All of Us data set in my talk. That currently isn't complete. There's around 330 individuals from the US in that resource with the linked genotypic and phenotypic data, which is still really good. It's still kind of the gold standard for the study of US healthcare data. But when that's complete, that will be 1 million people, which, obviously is substantially better than 300,000. So that's still a work in progress. Another part of that effort is, as I said, to try and improve the representation among groups who've typically not been studied in so much detail. So genetic research over the last kind of 10 to 20 years has predominantly focused on white Europeans. Because that's where a lot of the data was, that's where a lot of the methods were being developed. It completely unfair, of course, but that was just how the data was skewed. And now, it's beneficial to everybody, and it's more equitable if the research is performed in other populations. And that's undergoing at the moment. In terms of kind of other things that are being developed in the biobanks, I think moving beyond just having genetic data, so things like having proteomic data and having this expression data set can help to, because if we have someone's genetics and we have someone's healthcare records, we don't really know what's happening in the middle all the time to lead from the genotype, which is static, to the phenotype, which can be transient, or it can, it can manifest in a number of ways. So I think having more layers of data in there will really help to get to the bottom of what's happening. And, these data sets aren't necessarily tailored specifically to rare diseases, but in the UK, we work with the 100,000 Genomes Project, which is a rare disease and cancer data set. And there are more rare disease. I think there's a big rare disease effort underway at the moment in the EU to provide a larger data set for study as well.
Absolutely fantastic. And I know that there's some new JCA regulations regarding rare diseases as well. So hopefully that all helps the rare disease community a little bit more. Okay. The next question here is for you, Patricia. This question is, can you elaborate on projective techniques in qualitative research for rare disease? What is it and how does it differ from traditional qualitative research techniques?
Right. Well, traditionally in qualitative research, we introduce a series of topics, or we ask direct questions. Projective techniques are really designed to get below the surface of what's top of mind, because when you ask a direct question, you typically get whatever is top of mind. So if you want to get a little bit deeper, projective techniques using things like visuals can really help to understand some of the deeper insights that you need to have. So basically that's what a projective technique can do. And so that's why I'm very passionate about it, because I do think it helps. And also oftentimes the pre-work helps communicate very well to other people about what the experience is. So.
Mm-hmm. And just to follow up on that, since you mentioned pre-work, do you ever find that pre-work can skew the way that people report their disease symptoms or struggles in terms of terminology?
Not so much, no, because they're individual, they're individual exercises, and I think actually it would be the opposite, that you're getting each individual's feelings. It's less biased. Let's say you're in a focus group and you're presenting your particular visuals, then I think that there is that you're going to get each person's point of view much clearer than if you just ask a general question and everybody starts talking, and then there are usually some people who are try to bias the conversation. So I really like using these visuals because they do represent the individual pretty well.
Okay, fantastic. Thank you. The next question here is for you, James. This question is, we've talked a lot about establishing accurate figures for current prevalence. Are you able to estimate in future trends how any particular condition is likely to evolve over time and take into account any interventions such as earlier diagnosis?
Yeah, sure. So at Health Lumen, we use a technology called microsimulation a lot. So we use that for projecting an individual level population through into the future. And in doing so, we're able to account for things such as earlier diagnosis through improved screening. We're able to simulate the effect of new therapeutics. And that can all be kind of reported back as either like financial burdens and overall burden of disease, or kind of improvements in quality of life. So yeah, we are able to see what would happen for a given disease, both common and rare, given a certain intervention.
Okay, awesome. And then we have a few more questions here. So this next question is for you, Patricia. It's asking, do you have any thoughts on novel ways that patient experience data might be used in the future?
Yes, I think that what we may see in the future is, first of all, patient experience data today is mainly qualitative research, and don't think of it as really describing the phenotype for a rare disease patient. In other words, I think there's going to be this leap forward in understanding that patient experience data is really phenotypical descriptions of what is happening for an individual patient in rare disease, because we know there's a lot of heterogeneity. And so right now, we don't know what to do with that. But I think that it'll become clear how to deal with that in the future. And today we have patient experience data over here, and we've got disease models over here, and understanding the genotype that goes with this phenotype. And so I think of this world where we stop talking about patient experience data and we talk about phenotypical information, and then we marry that with this growing understanding of the genotype in such a way that we can understand each individual patient, what's happening in their what's happening to them, and therefore we can design better treatments. So all this work in rare disease can really drive forward precision medicine is where I want to go with this. So yeah, I think it's very exciting. And yeah, so James's world and my world might just come together more effectively.
That would be awesome. James, you'll have to make a database of patient experience data now.
I mean, that would be great. I mean, everything that we do is in silico, right? It's one side of the story. Getting the actual patient input and the patient experience is hugely important.
Absolutely. 100% agree. These two talks go really, really well together on that front. So I really wanted to thank you both for your expertise, sharing what your companies are up to, what you guys are up to, and how you're helping this community. Thank you so much for being here and taking the time to share with us. We really appreciate you. In closing, we hope that you enjoyed this Scientist.com and HealthEconomics.com cutting edge conversation sponsored by Health Lumen and ICON, and I hope that you enjoy the rest of your day.